Good morning, grade 12. Today we will start with a new section about human genetic disorders. It's section 14.2, part 1. The objective of this part is to determine how do small changes in DNA molecules affect human traits. We all know by now that genes are made up of DNA that interact with the environment to produce an individual's characteristics, or in other words, phenotype. However, when a gene fails to work, for example, in mutation, serious problems can result. Molecular research techniques have shown us a direct link between a phenotype and a genotype. For example, earwax can be of two forms, wet or dry. People usually of African and European ancestry are most likely to have a wet earwax, which is the dominant form. Those of Asian and Native American most often have the dry form, which is recessive. A simple base change from adenine to guanine causes the protein to produce dry earwax instead of wet earwax. Changes in a DNA sequence, like what happens during mutation, can change proteins by altering their amino acid sequences, which may directly affect one's phenotype, or in other words, the molecular basis for genetic disorders. Disorders caused by individual genes. Thousands of genetic disorders are caused by changes in individual genes, some of which we will be discussing sickle cell disease, cystic fibrosis, and third, Huntington's disease. What is sickle cell disease? Sickle cell disease is a disorder caused by a defective allele for beta-globulin. Beta-globulin is a kind of protein in hemoglobin. What is the role of hemoglobin? Hemoglobin usually carries oxygen in red blood cells. This defective polypeptide or protein makes hemoglobin a bit less soluble, causing the molecules to stick together when the blood's oxygen level decreases. As you can see in the following figure, that a mutated DNA replaces adenine with thymine and causes the protein to change to valine. This simple gene change caused the disease sickle cell. This sickle cell disease will clump molecules into long fibers, forcing them into a distinctive sickle shape, as you can see in the following figure, which gives the disorder its name. Sickle-shaped cells are more rigid than normal red blood cells. Therefore, they tend to get stuck in small capillaries, which are the narrowest blood vessels in the body. If blood stops moving through these capillaries, damage to cells, tissues, and even organs can result. Let's move on to cystic fibrosis. It is a disorder caused by a genetic change almost as small as the change in the earwax allele. Most cases, they result from the deletion of just 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator, or in other words, CFTR2 malfunction. What is the role of CFTR in cystic fibrosis? It usually allows chloride ions to pass across membrane. The loss of these 3 bases will remove a single amino acid, phenylalanine, from CFTR, causing the protein to fold improperly. With the cell membranes unable to transport chloride ions, tissues throughout the body will malfunction. Cystic fibrosis is a recessive allele, characterized by serious digestive problems and the production of thick and heavy mucus that clogs the lungs and breathing passageways. In this figure, you can see a comparison between a healthy trachea and a trachea that is clogged with mucus due to cystic fibrosis. The third and final genetic disease is Huntington disease. It is a disorder caused by a dominant allele for a protein found in brain cells. The allele for this disease contains a long string of bases in which the codon CAG, coding for the amino acid glutamine, repeats over and over again more than 40 times. The symptoms of Huntington's disease are mental deterioration and uncontrollable movements that usually do not appear until middle age. The greater the number of codons repeated, the CAG, the earlier the disease appears and more severe are its symptoms. This is the end of part 1 in section 14.2. Thank you for the attention.